The evolutionary biologist and author Richard Dawkins wrote an analogy to help us understand our own genetics. He compared our body to a large building with many rooms. Each room, he said, was like each cell in our body, and in every room is a bookcase. In his analogy, the bookcase is a nucleus, carrying 46 copies of the architect's plan for the building, much like our chromosomes carrying the plan for our cells. Every page of every book is a gene, and each word is written in the alphabet of A's, T's, G's and C's. And the analogy works to some extent, but Dawkins wrote it in 1973, and our knowledge of biochemistry has come a long way since then. Actual genes wouldn't be present on every page. They would only take up about 2% of the 46 volumes. And our cells are not as static as an empty room with a bookcase in it. It's a dynamic flow of information. So, for our purposes, we're going to have to extend Dawkins' analogy a little bit. Imagine in every room of the building there's an architect, reading their plan and maintaining the room constantly. This is a very general analogy for the central dogma: DNA to RNA to protein. Let's say the architect reads a page that tells them to build a window. Genetic diseases arise when that window can't be built. Diseases can have many etiologies, but, as is the case with the architect's window, they're usually due to a missing or non-functional protein. But what if there was a factory downtown that could mass produce a replacement window? This is gene cloning. Biochemists really have made these little factories that churn out proteins for cells, and hosts like E. coli are the machines. The gene for the necessary protein is inserted into an expression vector, for example a plasmid, which is then transformed into a host, often E. coli. The plasmid can replicate in the E. coli, and the E. coli are cultured so that lots of protein can be produced with the E. coli's protein synthesizing machinery. These E. coli machines have been used to overproduce insulin, which is isolated, purified, and injected into patients with diabetes. Another use of gene cloning is gene therapy, which involves adding genes, not proteins, to the cells of people with defective genes. In cystic fibrosis, a mutation in the gene for CFTR means a chloride ion channel, important in making sweat, digestive juices, and mucus, is non-functional. This leads to an accumulation of sticky mucus in the lungs. In gene therapy, plasmids containing the wild-type CFTR gene are delivered to the lungs of cystic fibrosis patients. Therapy has proven effective in trials, but gene therapy still has a long way to come, with the potential to treat over 6,000 genetic disorders. While it has clinical significance, gene cloning is very important in the biochemist's lab. We often need large amounts of protein for analysis and crystallization. This practical will teach you the fundamentals of gene cloning, so it's important to keep in mind its many uses.